Hello everyone. Today we will learn about SeqKit. This is a powerful toolkit to analyze FASTA or FASTQ files. SeqKit includes many subcommands for basic operations such as to translate a DNA sequence into a protein sequence or to generate statistics on the FASTQ or FASTA files. Also, we can use SeqKit for file format conversion from FASTQ to FASTA. Also, there are operations we can use to edit the sequence file, also to search the sequence file by sequence id or by a substring of sequence. Also, we can use order commands to sort or shuffle a FASTQ or FASTA file. There are also options for set operations. We can split the sequence or remove the duplicates, or to identify the duplicates we can use SeqKits. If we go to SeqKit's website, we will find a list of all the subcommands that are available within the SeqKit toolkit. So for each subcommand, there is a function, what it does. In most cases, the input is FASTA or FASTQ files. And there are many subcommands that can perform different tasks. If we click on the subcommands, that will show the detail of the function, what it does, and how to use these subcommands. So I would recommend you to look into all the subcommands, then you will find the commands that you need for your own analysis. Now let's go to terminal to install SeqKit into our work environment, as well as we will show some examples how to use SeqKit subcommands. Let's install SeqKit by copying and pasting this command. The SeqKit installation was successful. Now we will create an example FASTA file using the echo command. So this echo command is printing different lines in FASTA format. And let's save this file as an example FASTA file. So this is our FASTA file. We will use SeqKit subcommands to analyze this FASTA file. At first, we will use SeqKit's stats command, and this will give us the statistical summary where it is telling the file name example.fasta, format is a FASTA format, SeqKit can detect the file format, and type is DNA, number of sequence is 4, total length is 49 base pair, minimum length is 8, average length of the sequences, maximum length is 19. So this will give us an overall high level summary of the FASTA file. Now let's take a look into the sort command, how we can sort a FASTA file using SeqKit. We'll use the same file. So this sort command has sorted this FASTA file by sequence ID. The first one is SeQ2, then SeQ1, SeQ3, SeQ4. So this has not been sorted as I expected because there are differences in cases. These are uppercase sequences and these are the lowercase sequences. We can use I to ignore the case sensitivity. Now this sequence has been sorted by IDs and by ignoring the case sensitivities. So now the first one is SeQ1, then SeQ2, SeQ3, and SeQ4. Let's take a look into the other options while we want to sort our file. It says we can use different flags, such as we can sort by basis, by length, by name, uh, full name instead of just ID, by sequence. We can sort the FASTA or FASTQ file using those flags. So let's take an example how we can sort by sequence length. If we type -L, this will sort the file by sequence length. The smallest sequence is at the top, and gradually we can see these are sorted by sequence length. So using the command line interface, you can perform many other analyses using SeqKit. And SeqKit can also be used using pipe command. Let's take a look. Example, we can also use SeqKit stats. This will provide the similar output. So using pipe, we can also run SeqKit commands. Thank you for watching this tutorial. I hope you learned how to use SeqKit, and please find out more courses on my website. See you in the next one.